Hi, Jantine. Thanks for joining us at the last minute.
Hello, hello. Yeah, that was it must have been some I don't know telepathic
Exactly.
intuition to
Yeah.
write you.
Good. Good. Excellent. Yeah. Apologies about the last minute invite. It's
No, no, no.
crazy here.
It was good because otherwise I would have been on the T or something.
Got it. Got it. Okay. Okay. I think we have most folks, and since Mike might be late, why don't we just dive in? So as I mentioned, there's a couple of new names on the call here, so I thought we could just go around and give a 60-second or shorter intro to who we are and which project we're working on. For the newcomers, this is our every other week meeting to try and keep all the jointly funded projects between our group here at UVM and Mike's group there at Tufts aligned. We now have 4 funded projects at work. So there's a lot going on. And now, especially because the Mombot project is very big, we might have to fish in this meeting. We'll see how things go. But I obviously know this time works for Mike and most of you on the tough side. So that's why I sort of added Mombot to this meeting time. Okay. I'm Josh. I'm a computer scientist. I'm interested in evolutionary robotics and more recently biobots. And I'm the co-PI on 2 of the 4 funded projects we've got and the PI on 2 of the others. Fortunately, unfortunately, I'm the PI for the new Mombot one, which means I need to do all the paperwork and all the reporting. So I'm going to force some paperwork on all of you today in 5 minutes after we've done introductions. But it will be light, I promise. We got a lot of people on this call, so maybe we'll just do this in alphabetical order. Ali, I see you literally have your hands full. Can you introduce yourself for a moment, please?
Hi, I'm Allie. I work with Kai. I'm on the Xenobot project and I'm also a student at Tufts.
Great. Atusa?
Hi, I'm Achisa. I recently defended my thesis. I was working on unconventional computing with metamaterials, and I've been always interested in biobots. So I asked Josh if I could join these meetings from now on.
Thank you. Caitlin?
Hi, my name's Caitlin. I'm a PhD student with Josh, and I've most recently been working with Thomas and Pi on analyzing calcium signals in xenobots that are undergoing puncture damage.
Thanks, Caitlin. Doug?
Hi, I'm Doug Claxton. I'm a co-I on the Mombot project, and actually we have 5 proposals because I also have an NSF that I am the PI on with Sam Kriegman at Northeastern University or Northwestern University. And so I do all the bio end of the biobots, making small, medium, and large size varieties, and we'll be doing the sensing aspect of the Mombot project here in the lab.
Great. Thanks, Doug. Krishna?
I'm a PhD student with Josh. I've been working with I think, Sloan Xenobits project.
Oh, six. The Sloan project with Harvard. That's right.
Yeah, so I'm applying AI models to understand how Xenobots self-organize directly from videos. Yeah.
Great. Thank you. Piper?
Hi, I'm Piper. I'm Josh's PhD student as well. I work with Gizem on Anthrobot work with Caitlin, and I also work with the Yale team on metamaterial work.
Awesome. Thank you. Sean?
Hi, I'm Sean. I'm a postdoc in Josh's lab in complex systems, and I've been working on granular metamaterials, developing some of the work that Atusa started, and I will be attached to the Mombot project.
Thank you. Thomas?
I'm Thomas. I'm a postdoc in Josh's lab and also in the complex systems program. I am working with Caitlin on characterizing information processing in xenobots.
Awesome, thank you. And last but not least, Pi.
Hello, I'm Pi. I'm a research scientist in the Levin lab on the biology side. I've sort of 1 feet into not I shouldn't say departments, two areas. Background is like one area is developmental biology, but the other area is trying to understand basal cognition using the Xenobot models. So that's sort of the general area I work in.
That's great. Jantine.
Hi, I'm Jantine. I'm a philosopher and ethicist at the Wyss Institute, and I've been working with Mike and Josh and Doug on various projects. And yeah, I'm excited to be here. We finally became reality, the Biosense Mombot project.
Absolutely. We're glad you're here. We're glad it's a reality. It was a long time coming. So yeah, just to orient folks. So myself and Doug and Mike and Jantine are the 4 PIs on the Mombot project. So the 4 of us are on the hook to report back to Corel. Luckily, none of you have to do any of that. So obviously Mike's not here. Is there anyone else from the Tufts side that's missing today? Can someone from Tufts introduce or describe those that are usually here but couldn't be here today?
Yeah, I think Elena is a postdoc in our lab or staff scientist who's working on the project of looking how bacteria and xenobots can pass information across kingdoms back and forth.
Yep, great. And Gizem is probably off building anthrobots, I would guess. And I know there are 2 folks in the Levin lab that are working on the new one, the Zap GPT project. Can someone introduce them, please? Just so we have it on the record here. Maybe Thomas will have to do this one.
Yeah.
Yes. I was on Zap GPT as well, which for people who don't know is a project sort of unrelated to Xenobots involving using LLMs like ChatGPT to serve as a translator so that human scientists can somehow communicate directly with cells. And I was on that. On this side, I'm moving over to Mombot, but the 2 guys in Patrick's lab, in Mike's lab, are Patrick Erickson and Yanbo Zhang, and I've met with them once. They seem like great guys, but I couldn't tell you much about them. But they're out there somewhere.
Okay. At least we've got their names on the recording here. That's great. Thank you. On our side, we are going to have 5 members of my lab working on the Mombot project. 4 of them are here: Krishna, Piper, Sean, and Thomas. The 5th member, Amanda, I don't think anyone in the Tufts folks in the Tufts side have met Amanda Birchinger yet. She's been working on another project, which is finishing up. So she's going to be rotating onto the Mombot project. And I just completely forgot to invite her to this meeting. So that's why she's not here. But she'll be able to watch the recording and hopefully she can join us for the next meeting. I think that's everyone. Are we missing anyone? Okay. Other people or institutions we should be aware of. I just mentioned the 4 PIs on the Mombot project. Tufts is going to be working very closely with Boston Engineering. Doug, do you want to just describe who Boston Engineering is, generally speaking, what they're going to be doing?
Yep, Boston Engineering is a fairly large company that's located just outside of Boston that we have had some agreements with in the past. They've built us some automated behavior devices for tracking aquatic animals and applying electric stimulus, and they've been great partners through those projects. And so when we came up with the Mombot project, that was one of the first companies we reached out to. So they are going to be responsible for the lion's share of the money and deliverables, I believe, which is creating a custom device that will basically apply stimuli to cells to have them assemble in various ways. And those stimuli we'll talk about later, I'm sure, in detail, but combinations of temperature, electricity, chemical factors, etc. It will incubate different types of cells over different time courses, and then also observe the behavior of those cells through custom optics solutions. And so that will be probably, I think, a 2-year plan involving a number of individuals there. And our goal is to have weekly or bi-weekly meetings with their folks to make sure the biology, the computational end, and the hardware are all going to align. And Josh, if you want to comment too, I believe the second dovetail there is the AI that will be built through the Vermont team will talk to MomBot to input signals and understand what's coming out of the back end to create a closed loop system where it can understand how various perturbations affect the cell geometries and behaviors that come out of what MomBot is cooking, so to speak.
Awesome. Yeah, thank you. And I want to actually, when we finish up all the intros here and orientation, I want to actually talk exactly about this, because there is now a meeting on the books with Boston Engineering, and the next one is this coming Tuesday morning, right, Doug?
Correct. It's there. Just to frame with the group as well, these Boston Engineering has been very good partners with us, so I'm excited to work with them again. But in our early meetings, it's clear that they also function as a business, so we want to make sure our strategic interest in the project are met, whereas we want them to do everything, and I think their goals is to commit to as little as possible. So we will be searching for the best compromise there.
Exactly. So I know in this meeting we usually just talk basic science, but if you'll forgive me, we'll make an exception today. I want to make sure that at least Doug and hopefully Mike can go into that meeting Tuesday morning well armed to represent our interests, to make sure we can do the kind of science that we want to do. I've got sort of a vague idea about how to do that, but I want to make sure we all brainstorm on how to actually do that. And that's, I think, probably a very good use of our time today. Yeah. Okay. So yeah, I actually have a picture of what Doug just described and timelines, and I want to review them. I also am going to put on my principal investigator hat here for the MomBot project and issue a couple of disclaimers, things to keep in mind. The MomBot project is being funded. The money is coming from a national lab, CREL. I'll put the acronym in the chat just so people have it, which is based in New Hampshire. CREL in turn got the money for the MomBot project from the federal government. There was a federal appropriation. So this is federal taxpayer money coming through Department of Defense for us. So this is a Department of Defense project. It's obviously an unclassified project, or else we wouldn't be talking about it on Zoom. But it's important to just keep in mind where this money is coming from. So for those that are directly going to be working on this project, if you handle any file, any cell, anything to do with the MomBot project, let me know, and you've got to read some rules and regs about what you can and can't do. I don't know why we got a specific email saying no computer can be involved in the MomBot project on which TikTok is running. So if you're a TikTok user, bear that in mind. I laughed also, and then wasn't laughing. So, again, it's just a good reminder that although it's an unclassified project, we generally speaking, we'll try and do our best to keep whatever we discuss amongst ourselves, and we should probably be okay. Another good rule of thumb that I learned from my advisor. Oh, sorry, Thomas, did you have a question?
Yeah, so I know we can't have TikTok installed on our computers, but there's people post videos and links like to social media all the time to TikToks. What is like the level of exposure here that is considered safe?
Exactly. I don't think anyone knows anymore, right? Usually you would download TikTok and install it on your phone or your laptop, but now so much stuff is in the cloud. What counts as, I don't know. I don't know if anyone knows. So if you're curious, again, I can send you all the documentation and you can read through all the nitty-gritty details to make sure that we're in compliance. Jantine.
I think everybody has been thinking about this, but given the fact that this is a Department of Defense project, Anyone, of course, who has personal objections or difficulties with being involved with, such a funding source, I think they should feel free to talk with Josh about it.
And not just me. Talk to Doug, talk to you,
Yeah,
we
but
all have perspectives on why you may or may not want to be involved. What does it mean to be involved? Yeah, absolutely. I think, at least on my side, for many of us, this might be your first DoD project. It's good to talk about.
yeah, and for people who are not US citizens, depends from what country you come and what your immigration status is in the US. I think you need to, I've been involved in such a project before, and then asked, that was DARPA, am I working for the US military as a Dutch citizen? And then the program leader said the answer is yes, which technically would be a problem with the Netherlands, but practically not, because we are like allies or so. But this may be for other countries like different. So I think you should at least try to consider your own situation, and there will be a solution, but it's bad to get a surprise afterwards.
Absolutely. Thanks for that, Jantine. Yeah, absolutely. Think on it and let us know. And if we don't know the answer, there's several groups on campus that knows how to navigate these waters better than we do, and we can point you to those groups. Okay. Let's see. Anything else to mention about that? Oh, so I mentioned to my own lab on Monday about this new project. If folks ask, if you're actually funded on this project and ask you, what are you working on? Again, the advice I got from my advisor was give, sort of the one sentence answer. I'm on a funded project to continue the Xenobots project. And the Xenobots work here, it's all been published. We're continuing on with that work. Just, common sense applies here. Keep things vague and concise, short, and that's usually good. Another rule of thumb, again, is as we get going, there's going to be lots of files that are sitting on the cloud and sitting on different computers and biological materials and all the rest. A good rule of thumb is at any point, if Doug and Jantine and Mike and I have to be involved in a review with Krell, or someone from Department of Defense comes and says, where are all the materials? We should be able to generally point to where they are. They're on these computers, they're on this shared folder, they're here. And if Krell says, is that everything? And we say, no, we know there's some additional files, but I don't really know where they are. That's not so great. So just generally speaking, you can sort of check in with yourself. If someone were to ask you, would you be able to point to where everything is? Or point to someone who knows? Again, as long as you don't know, if you can point to, if any of the UVM folks, you can point to me, any of the Tufts folks, point to Doug or Mike. And obviously Doug, Mike, Jantine, and myself, we should be able to know where everything is. Okay. Any other? Yep.
Can I add one thing on the bio end? There's only a couple bio people here, but for people working on bio things or computational folks that are maybe thinking about the bio end, we'll have very direct objectives. Then there's always room to meander, but this is a project where we need to be especially careful. There's some very specific nomenclature in the rules about generating any biological material that is more resilient in the environment than it would be naturally. So more resilient to temperature or antioxidants or chemicals. And so feature creep in this is fine, but just make sure that you flag any changes in goals to the PI so that we can approve it, to make sure that we don't accidentally run out of compliance with what the government has supplied us.
Yeah. Great point, Doug. Thank you. Okay. All right. I'm going to share my screen and we're going to just look at some paperwork for a moment. Again, my apologies. So I have made a master doc, and again, in the spirit of maintaining a good paper trail, so we know where everything is. I'm not the best spreadsheet maker, but here's my best attempt. So for those that are on the project, I want you to bookmark the link for this spreadsheet. Here's the link right here. Can everybody see this okay? Please write down or record this URL. This will point you to this master spreadsheet. Thank you. Thanks, Doug. Okay. Okay, there's a lot of detail in here, but again, if you have any questions about where we are, what we're doing, there are links inside this spreadsheet to the original proposal. There's the cost narrative, so there's a very long document that we sent to Krell that says how exactly we're going to spend all the money, and the budget is a summary of all of that. You can already guess a lot of the details inside these 3 links. These are definitely not things we want to share beyond this group and those that are immediately working on this project. So like anything on a funded project, but specifically on a DoD funded project, think before you share a link. But if you're working on this project and somebody else is working on this project and they don't remember where so-and-so is, send them this link, point them where inside this link they need to go. I really want to try and minimize, emails with attachments and Word documents and all the rest. I know depending on what we're doing, we might need to write things in Word documents and PDFs and share them as attachments. But if we don't need to, let's not. Let's try and link it inside this document hierarchy. So again, we can keep track of where everything is. Make sense? Okay. In the spirit of maintaining a good paper trail, you'll notice there's I just added a second tab to the spreadsheet just before we met. And I am going to try and do my best of keeping a running record of every meeting that's held in which MOMBOT materials are discussed or work is discussed. So Tufts folks, if you meet with Boston Engineering or you have an internal meeting or anything that you think is worth adding to this spreadsheet or this tab, please add it. Jantine, if you reach out to some members of the team and you want to learn more about what they're working on, same thing, just note it down here. Here's today's meeting that we're holding right now. When we finish up this meeting, I will post the recording and drop a link in here, and I will restrict who can view the recording of this meeting. So again, we can point to, we've got a good record of everything. Again, I'm not the best spreadsheet maker, so if people have better ideas about how to make sure everything is centrally located, I'm all ears. Okay. The other reason I want to make sure that we have a very careful paper trail is we're going to have some program reviews with Krell, and I don't know yet what they're going to ask us for. Doug, we've been through this before with DARPA. You never know what they're going to ask for in a report. So this is why I want to make sure everything is written down. And if it's not written down, it's in some Zoom recording somewhere. We can go back, have a look, pull out the transcript. Again, we probably can't do a perfect job at this, but as long as we do due diligence, I think we'll be in good shape. Other reason for maintaining a good paper trail is we want to make sure that Jantine is successful in this project. Jantine, for now, your job, as far as I understand, is to learn what we're doing and observe how this project goes from day one. Today is, in effect, day one. So hopefully this will be a good resource for you if you want to go back and use this as archival materials or whatever.
Yeah, and I think, Josh, people may have seen in my, I think it's in the green part of the spreadsheet, like, what am I going to do as the ethicist on the project? I'm basically going to like ethics assessment. That means concretely, I'm going to talk with everybody involved in the project, and that's in groups, and for the 3 phases of the project. So at some point, or maybe already soon, because we started today, and I will approach people, and it can be individually, or we can have like small group meeting where I will raise discussion topics that I still need to think of which ones. But so be tuned, because I may reach out, and then it's probably as you say now, Josh, it's via this spreadsheet.
Please, yes. Thank you, Jantine, for orienting us to your part of this project. I think, again, coming back to everyone taking some time to think about whether they want to be involved in this project. There's many reasons why Jantine is a part of this team. From my perspective, one of the reasons why is I want someone, a little bit removed to watch what we do from the very beginning to help us think through, what are the not so obvious repercussions of, what we're trying to attempt in this very large project. Wouldn't it be terrible if we got to the end and say, shoot, it would have been great if we had another pair of eyes looking at what we're doing. They might have helped us see in advance some of these unexpected, repercussions of this work. Okay.
Yeah, but I will also, of course, look not only at, say, negative implications, but also at positive implications.
Yes. Sorry, I didn't mean to paint you as our watchdog. Yeah, but both.
No, I'm not an oversight person. I think that should be
sorry.
like also in the weeds. I'm not in any form oversight or so. I'm a colleague and a member of the team. So
Absolutely. Thank you, Jantine. My apologies. Okay. I'll just pause for a moment. Questions? Okay. All right. So let me just try and walk you around this spreadsheet so you can get a general sense of what we're doing and when and who and so on. So yeah, it's as Jantine said, it's broken into red. Everything in red is UVM's responsibility. Everything that's in blue is Tufts' responsibility. Everything in green is Jantine's responsibility. As Jantine mentioned, you'll see that for each of the three groups, there are specific things we have to get done in phases. This is year one, this is year two, and this is year three. And you'll notice that there's three nouns that are repeated in all three groups. Year one is biobots, year two is biosensors, and year three is excretory biosensors. I don't know if this necessarily makes the most sense about how to distribute the work over 3 years. This was the best thing that we could come up with at the time. So biobots basically means xenobots. Doug, correct me if I'm wrong, but I do not think anthrobots is going to be a part of MomBot.
Correct.
Okay. All right. So wherever you see biobots, that basically means xenobots. And the reason that biobots is mentioned in year one is the assumption is we're not going to be changing the xenobots too much in year one. It's mostly automating all of the things that we had to do manually here at UVM and you all had to do manually at Tufts when we did the original xenobots work. So 10,000 foot view for year one is try and automate as much as possible. Human hands off. And we can again talk about how to do that. That's basically the idea. Doug is working on turning the xenobots into biosensors. Doug, do you want to give a 60-second summary of what that means?
Yes, I'm still trying to figure out what this means. So the very short version is one thing that we don't have a great ability for with the biobots is to understand the signals that they can sense and how they might be able to advertise to us either obviously or cryptically what they're sensing. So the hard part of this project is Krell was very tight-lipped about what they want us to sense and respond to. And every time we gave them an idea, they said, try again. And we went around about 5 times. And so what we settled on, although I think there is some room to navigate if we find something interesting here, but initially, at least, we're going to be looking at cues that are released by naturally occurring bacteria that would be in aquatic ecosystems. So can they sense different bacterial compounds? And can they differentiate between different bacterial families? And also reactive oxygen species. So these are a very common environmental pollutant that one form that you might know is hydrogen peroxide. These are very nasty chemicals that overrun the antioxidant capabilities of cells and kills them. But it's produced by a lot of naturally and non-naturally occurring chemicals in the environment. We chose that one as the very short version because there are already transgenic lines of frogs that have reporters that are built in that sense peroxide and reactive oxygen and turn on fluorescent molecules. So in that, we would be looking at how sensitive is that circuit? How long does it take for the signal to come on? How long does it persist? All of these are really simple questions that I think in a worst case scenario, we can generate really good data for to give to the AI.
And when they react or they sense it, Doug, it would be that there'd be some change that's internal to the xenobots, something inside them, right?
Correct. They turn something on. So in the earlier case of bacteria, it is these antimicrobial peptides that we're hoping to sense. In the case of the genetic circuit, they turn on an actual GFP molecule. So they start glowing when they sense the reactive oxygen in the environment.
Great, thank you. So that's what we mean by biosensors in year 2. Doug, do you want to talk about what we're going to try and automatically design and manufacture in year 3, the excretory biosensors?
Yes. So following up on that, getting them to produce something is step 1, but we want to be able to observe it. So having them release something into the environment that we can detect with either a chemical test or something just optically. They change the color of the water from clear to purple, for example. So it's really a detection method that we have that's either obvious or cryptic. And I believe that on the AI end, we would look to see if the AI can discover variables that will enhance this excretory response to make it more or less sensitive to the compounds of interest.
Thanks, Doug. So we're spreading things out over the 3 teams and then spreading things out over these 3 years, where we're basically going to repeat more or less the same process in each of the 3 years. We're going to try and automate the design and automate the manufacture of X, where in year 1, X is traditional xenobots, in year 2, biosensors, in year 3, excretory biosensors. So far, so good? Any questions? Okay, so I guess I won't go, I don't think we need to go delve down into this much detail. I'll just say very briefly on the UVM side, we've further broken down each of our years into 5 little work packages, and those correspond hopefully to the 5 folks on the UVM side. It's not necessarily something we need to do, it's just something we did on the UVM side, so it's clear who's doing what. And I think it's too much detail, but maybe on a later meeting, we'll review and go over how all these work packages fit together and how we're going to handshake each of them with what's going on at Tufts and what Jantine is trying to accomplish in this project. Okay.
Josh, one question on the comp side. As you develop software, the way that we have distributed this in the past is through GitHub. Is that going to be a problem with Krell? What is the mechanism from which you would like to be able to pass us code?
Yeah, exactly. So I definitely want to talk about that today, because I think you need to lobby for that with Boston Engineering, like starting Tuesday morning. So we got 25 minutes left. I definitely want to leave time to talk about exactly that. How are these files going to go back and forth between UVM and Tufts? It feels like a super minor issue, but it's not. Good question. We'll come back to it. So that's a little bit about how things are spread out over time and who's doing what, generally speaking. And then again, our partner here is Krell. As far as I know, they don't care, and we don't have to give them anything until September 2024. So in the fall, you all at Tufts have to mail 100 biobots to Krell. Krell stands for Cold Regions Research and Engineering Lab. So their role in this project is to see how these things that we automatically design and manufacture handle cold regions. So I don't know what testing they're going to do. They kind of said a few things, but Doug, I agree with you. I think they're being deliberately cagey, and maybe that's a good thing. I don't necessarily want to know what they want to do with them, but yeah, we have to send. That's going to be our first engagement with Krell. The reason I wanted to point that out as the first engagement with them is we are on a fixed price contract with them. This is a contract not a grant. Most of you have been on grants, written grants, been funded through grants. A contract is exactly what it says. It's fixed price. And CREL has the option to withdraw the money basically at any point. So in order to keep this project running and the money flowing, wherever there's an X in this spreadsheet, whenever there is an X arising in this spreadsheet, please make sure if you're responsible for it or involved with it, that we make that deadline. And if you're unclear what needs to be done and so on, talk to Doug, talk to Jeantine, talk to Mike, talk to myself. But the first one is mailing 100 Xenobots to CREL in September. Okay, as you'll see, most of the things we need to deliver to CREL are in 12 months from now, so the end of March. There will probably be a program review meeting then. I've been involved in a CREL project before, and that's usually how things went. There was a lot of flurry of activity at the end of each year where the performers, which is us, had to demonstrate we actually did what we promised to do. And CREL, like many national labs, is a stickler for detail. They're going to read the fine print of the proposal and exactly what we promised, and they're going to make sure that we're giving them exactly that. So yeah, attention to detail here matters, and hopefully we'll spend good parts of these meetings making sure we're clear on exactly what we need to deliver to them and when. Any questions about that? Okay, all right, so yeah, exactly. Let's go back to what Doug was mentioning here. I think most of you have seen this picture before. This is sort of for CREL or for the higher ups to understand the basic idea here.
Hey, Josh, sorry, we can't see anything other
can't.
than the spreadsheet.
Okay, sorry. Let me try that again. How's that?
Better.
Okay, sorry. This is the figure that we prepped long ago for CREL and the higher ups. All the computational stuff is on the left and all the biological stuff is on the right. Generally speaking, we in panel A here are going to collectively come up with what we want the biobots or the biosensors to do. Like Doug was saying, we want them to react as much to hydrogen peroxide as possible, or whatever it is we choose. The loss function, the objective, the thing that we want them to do. Then the AI on our side here at UVM is going to search in panel C here. The AI is going to search over all the possible ways that it can think of to externally perturb the cells, to assemble as shown in panel D, and then do something in panel E. So the AI is trying to come up with a perturbation, and that's what we're sending to Tufts. The easiest way I can think to do this is that on our side, we run this on a laptop, and the laptop automatically uploads the AI's best guess about the best perturbation, uploads it to Dropbox, assuming CREL allows us to use Dropbox. Mombot has Dropbox installed, and then Mombot does its thing. It tries to actually execute whatever is in that file to the dissociated cells, which are shown in F. So there's F. There's the dissociated cells being perturbed. And that triggers or modulates somehow the self-assembly process in G and H, and or the behavior of the resulting multicellular assembly in panel I. Doug, did I get any of that? Doug or Pi, did I get any of that right or wrong? Good. Okay. All right. And then hopefully with none of you on the tough side touching anything, so Mombot would upload a file back into exactly the same Dropbox folder, which at the moment, unless anyone can think of anything better, is an MP4. It's just a raw video or videos from the cameras on Mombot. That's it. And then the AI learns whether or not, or how well the perturbation actually did at producing what we wanted the bot or the sensor to do in reality. Pretty simple. Okay, so Doug, I would say, unless you have a better idea, if when Boston Engineering asks, what's the minimal viable product? What does this prototype look like? This would be it, I think. What do you think?
Yes, I think that's good. And I believe you answered my question from the internal end. I was also just making sure on the external end that Krell and or the US government doesn't, they're so finicky about how files are uploaded and transferred. We may also just want to make sure that Dropbox is viable from a programmatic standpoint.
I imagine it probably isn't. So I'm going to email Krell and say, this is the plan for the moment. And they're probably going to squawk and we're going to have to update this.
Yep.
But I want to make sure that we're able to frame how this goes for Boston Engineering. If they say, well, our usual equipment, you need to do FTP and someone needs to type in. The moment you hear a verb like someone needs to turn a knob or someone needs to type in, that's the thing I think we need to try and nip in the bud as best we can.
Yep. And then on the bio ends for the bio folks, the optics here are still somewhat TBD, but there's a lot of things that we can measure. So fluorescent signals, optical density, movement, even just live or dead, right? There are some treatments that are going to just dissolve the cells. And we should be able to extract all of that data in an automated fashion to reduce the time that it takes the investigators to do this really labor-intensive process.
Great, thank you. So I've kind of deliberately made the interface between our group and your group like ridiculously simple. Upload and download one file in a Dropbox folder. Also to try and make it clear that, it would be great if we don't have to transfer back and forth, huge sorts of things, that we can literally do things in this very, very light manner. It'll keep things modular and it'll help both groups focus on the science that they need, the science and engineering that they need to do to get the work done without having to call you at Tufts every hour on Slack and figure out what's going on, that we can, try and work independently as much as possible. That's part of the idea. Anyone else have any observations? Thomas?
Yeah, could we possibly set up some kind of joint UVM Tufts Slack? Like you just mentioned Slack, and it occurred to me that while we don't want to be messaging each other every 5 minutes, it would be good to have a space to communicate that wasn't email or bi-weekly meetings.
Yep. Thank you. I'm glad you brought it up, and I don't have to ask directly. I'll just pass on your request. I know we all get burdened with Slack and Teams and Zoom and whatever it is, but I think it's probably worth it. Again, I'll have to check with Krell that we can, but is it okay with the Tufts folks? And Jantine, is it okay with you if we set up a MomBot specific Slack? Okay, all right, I will check, but yeah, I think that would make sense. Yes, we probably don't want to be slacking each other, messaging each other every hour, but this is also another thing to keep in mind as we try and frame this whole thing to our advantage so that we can do useful science and engineering, which is MomBot's going to be a real-time machine, right? Like when the dissociated cells are ready to be zapped by MomBot, MomBot better have instructions from our AI about what to do. It would be a shame if MomBot is sitting there twiddling her thumbs with not knowing what to do. And same thing, but to a lesser degree on the AI side. If it's got ideas, it's got ideas about things it wants to try, and it's got to wait a week or 2 weeks to figure out, to get an answer, what happens if you apply this field. So there's also, and it's way too early to do this now, but to start to think about timing. the Anthrobots project has been very helpful to me from an educational perspective because I now know there's Anthrobot seasons, right? So presumably there are going to be times when Mombot needs a lot of external perturbations like rapidly in a row, and then other periods where she can't take any new requests. What is that timing, generally speaking, gonna look like? Can she zap the cells once every week at a regular basis, or once every hour for a couple days and then nothing for a couple weeks? What is that going to look like? Again, I'm guessing it's probably way too early for that. Caitlin?
So this picture has the aggregation of the biobots, or like the formation of the biobots, and then the behavior analysis kind of in sequence. So I'm imagining that in the beginning of this project, we might want to like iterate over those two things, maybe in parallel. So like Krishna iterates over it, and Piper is iterating over it separately. So I'm wondering if Mombot will have that functionality to kind of do both of these things in parallel, or kind of what that would look like. Yeah.
Doug, do you want to speak to that?
Sure. Can you rephrase, Caitlin, just so I make sure I'm answering the right question?
Yeah, so I can. So Christian's kind of working on this AI model to kind of predict how the aggregation or the formation of the biobots will be affected, or how we can kind of control that. So I could imagine he would want to iterate through this loop up until H, and then kind of bypass I right there and get feedback from Mamba, and then go through this loop. So I'm wondering if that will be something that will be built into this kind of minimal model. And then Piper, on the other hand, is looking at how we can control swarms of biobots. We'll kind of be working on another level of this loop. So we'll want to get feedback on I, but maybe bypass what Krishna is working on. So instead of going through this loop all in sequence, we might want to be like developing different parts of it.
Yeah, so I think so. Two things. I believe in the original plan, the idea is that all of this is modular, so you can skip or repeat any steps that you want. So there may be some things where we don't actually care how the cells assemble per se. We're treating them with something so that the same ball of cells responds differently to different stimuli, right? So that might be prepping them by hitting them with temperatures at certain times. So you have a ball of cells and another ball of cells that have different cold tolerances. In which case we don't really care about the morphology. And there may be things where we only want the morphology to be impacted. And so I think what you're saying is correct. All of this, some things will contain all of the letters in alphabetical sequence, and some will have gaps, and some may even have repeated sections where we're doing repeated observations over time. And so I think it was written this way to be as general, as flexible as possible. On the bio end, I can also say that all of this it will take time for for Boston Engineering to provide us with anything useful, right? They have to build this, and that will take quite some time, I think, before we have a device that's running this. In the meantime, we can do this the old-fashioned way in the wet lab, which is by manual experiments. So don't let Boston Engineering hold up your computational end. If there's things that we can test, we may have to be more picky, but we can pilot and maybe figure out where's the best place to get the crowbar in to start getting good data for the reports. And we're happy to generate that data on the biological end manually.
So I think to that point, if I understand correctly, Pi, you're going to be Mombot until there's actually a Mombot, because you now know that an electric field actually does affect the xenobots, right? You are someone who's working with you. So my feeling is the best use of our time in the next couple of months is we'll generate an electric field exactly like you induced the electric fields when Princeton was visiting. Whatever that is, I don't know exactly the file format or what parameter settings you need. You tell us, and then our supercomputer or a laptop here will upload a file with that, and then the Tufts folks would apply it to cells and send us back the MP4. So we actually close this loop and document that process. And that's what Boston Engineering is going to want to observe or get a written record of, right? Okay. Pi, my apologies. I still haven't watched the recording, but Caitlin was telling me on Monday that the electric field didn't seem to influence self-assembly, but it did influence the cilia. Is that right?
Yes and no. We don't know about self-assembly because they were here only for a couple of days, so we were only able to run short-term experiments.
Okay.
So, the self-assembly part will require long-term experiments or long-term exposure. So in the short-term experiments, we did see that the electric fields that we were using were changing their movement patterns, movement behaviors, and that at least in part was because the way the electric fields were affecting the cilia motion or cilia movement or cilia assembly or cilia structure. So that part we have some confidence on. We have no idea about the assembly from dissociated cells because that would require applying current over maybe 12 hours or 24 hours. We just didn't have that much time.
Got it. Got it. So the current that was influencing the way the xenobots moved, did you zap the cells and then turn off the zap and then they acted differently? Or was it real-time control or whatever?
Yeah, it was the first one that you said. So the way we were doing it was documenting motion behavior prior to the current, then during the current, and then after the current of the same bot for multiple bots. And what we were seeing is that it moves in one fashion, and then when we started the current, it stopped or changed motion in some way, and then we turned it off. It after a little while, some of the bots regained their previous motion, the one before the electric current. Some didn't because their cilia were sort of destroyed on one half, so they kept moving in the same direction. So that's those are all the pilot experiments that we did. Yeah.
Okay, fantastic. So we have here on the UVM side, we're going to have our first internal meeting about this next week at some point. So I think what we're going to do on our side is develop the computational stuff around what you just said, that exact use case. So because it didn't seem to influence behavior in some of the bots, influence movement, we'll make a movement loss function, a movement objective. We're going to try and get the AI to dream up a way to apply current to produce some desired movement trajectory. Does that seem reasonable?
Yeah.
Great. Okay, awesome. So I think so for the software engineering side of this and system engineering, that's it. We just take this sort of seed of a use case and start to develop this bigger, a minimal version of this figure 8 around that use case. And even if we haven't, like Doug said, it's going to take a while for BE to build this stuff. But if we can do this go around this loop manually a few times, and I think on our side, if we're doing this completely manually, even at the end of this first year, we might not have enough data for the AI to be able to learn how electric fields influence movement. But if we could at least complete a few rounds and show that the AI is like getting better, it's reducing error, that I think would be at least satisfying to us and probably satisfying to Krell. We could tell a good story at the end of year 1 that we've succeeded. So I think, again, for now, just putting a stake in the ground, that's a good direction for us to head in, unless people have other ideas. Okay, cool. Well, thank you. That's much clearer in my head. Okay. All right. I'm sorry I did all the talking or most of the talking today. Anyone? We got 5 minutes left. Anything else? Mombot related or otherwise?
Can I ask a question of Pi that has nothing to do with Mombots?
Sure.
Okay.
And has there been, I realize I've missed a couple of these meetings for various things beyond my control. Has there been any developments on the mutant xenobots? We had talked about trying to do some calcium analysis to see if we could relate morphology and functional connectivity.
Yeah, I mean, no more experiments since the last time we talked. I'm working on a paper, finishing that part up. So mostly doing reading, writing at this point. So not since we've talked last time. I
Okay.
haven't done any more experiments on that.
Cool. Yeah, that's totally fine. I just wanted to
Yeah.
bring it both to the top of our minds just for a moment because I'm
Thank you.
still very interested in that.
Oh, me too. I'm super interested, but I need to get this paper out.
For Caitlin and Thomas, and I think Hannah's not here. What do we have like a penultimate version of this Xenomics paper finished? I'm happy to pull it offline and start putting in references, but our ability to edit it is going to be in sort of a master document format that we're sending around after that. Do we want to maybe send one more email to the PIs and say, put whatever you want in before we start finalizing it for submission? Does that sound good to everybody?
Yeah.
Sounds good to me.
Sorry, the last I heard that the cover letter was being prepared. Has that been done? What's the status there?
I've not seen the cover letter, but the rest of the text is now, I believe, intact and pretty polished. There weren't any holes anymore. There was just a couple small comments left, but I think I can resolve most of it.
Caitlin, Hannah, and I were talking about the cover letter as a way to kind of preempt, some of the criticisms that Olaf thought we could get from reviewers. I believe I volunteered to write that, and I confess I have not done so yet. But
I think Hannah had responded that she would write it, and I was in touch with her at the end of last week, and she said she was still working on writing that and was spending the rest of the week
that makes me
focused on
feel
that.
so much less guilty.
That's fine.
Okay.
Yeah, I haven't heard from her since. So yeah, I'm not quite sure what the state is, but I will keep in touch with her.
Yeah.
Anyone else have anything they want to toss out there?
Do we know if like the VAC is sufficiently secure to be running these things? Like what if there's another group importing TikTok onto the VAC?
Yes, I spent most of this morning emailing back and forth with the compliance office here at UVM at exactly about that. So luckily, we need to raise these questions and they've got the answers. So you can go ahead and run stuff on the VAC for now, and I will keep checking with them. If they think it's not a good idea, we will find an alternative venue.
On that same note, I have been, perhaps against the rules, still using IU supercomputing facilities because I have access for another year or two. Should I not do that for anything biobot related?
You should not do that for anything biobot relating because that's on directly or indirectly. It's on IU's dime and their resources, and that's not stipulated in the grant. IU is not mentioned anywhere in the proposal. So
okay.
Yes, please. Okay, we got 1 minute left. I just want to end with a personal note. The automated design and like manufacture of autonomous machines, for me, this is like a lifetime dream. My postdoc advisor, Hod, 25 years ago, he wanted to do this too. He's been working on it for 25 years. So if we're successful, he's going to have to buy me a beer. This is going to be very exciting. So there's going to be a lot of paperwork and red tape, involved in this, but I'm excited. This is I know a lot of roboticists, a lot of folks have tried versions of this in various ways, and no one's managed to pull this off. If we can upload a little equation that says what we want the bots to do, and a physical bot pops out the far side of MomBot and actually does that, it's going to be a huge, 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 huge deal, at least in the robotics community. Huge. So I am crazy excited about this. Thanks so much, everyone. I apologize for monopolizing the stage today. See you all in 2 weeks' time.
Congratulations for the grant.
Thank you. Thank you.